So new heading, new ink, left-hand column of your notes. You're going to put stages of cell cycle. So we've got 4 stages with different things happening in each. Some stages are more complicated than others. In your notes, beside stages of cell cycle, I want you to put that G1 is growth and just kind of normal stuff. S is DNA replication. So that's where my apples and trees and cars and garage, that's where that comes into play. We have to match it and make a new one. G2 is growth and preparation for mitosis. So G1 growth, normal. S is DNA replication, apples and trees, cars and garage. G2 is growth and prepare for mitosis. M is mitosis, which is where the nucleus divides. It's mitosis. It has 4 parts. They are prophase, metaphase, anaphase, and telophase. We're going to go into each one of those steps in detail today. Here's what you need to know. If we fail to go through one of our gaps, either G1 or G2, those are like our checkpoints. And if we do not respond to checkpoints, it leads to cancer. That's what causes cancer and tumors, is if we're not doing what we're supposed to in G1 and G2.
What are they supposed to do?
Normal growth and normal preparation for mitosis. But if we skip over one of those steps, we go right into replication, then all that cell is stuck doing is making another copy and making another copy and making another copy and making another copy. And that's how tumors happen. I think you'd still die, but I guess your family could be wealthy. Give me a fist of 5 on this. Okay, several minutes, that's fine. So as you're writing, I am going to review it one more time. G1 is when growth and normal things happen. S is where it replicates. G2 is, again, another phase of growth and preparing for mitosis. M is mitosis. If we miss G1 or G2, those checkpoints, that is when we can end up with cancer. All right. About to move on. Sure you're getting what you need right quick. Remember, guys, it's okay to abbreviate. It's okay to just write words and not complete sentences.
Not in English, though.
So we are going to look at each parts of the cell cycle now. So the first thing that we're going to look at, so new heading, new ink, left-hand column, I want you to write interphase. So change your pen, go back to the left-hand column, and write the word interphase. So interphase actually includes 3 of these steps. So interphase begins when a cell is created and ends with cell division. So there's actually 3 stages. We've got growth, replication, and preparation for division. That's basically G1, S, and G2. All 3 of those steps are a part of what we call interphase. Cell is created to right before it divides. It includes growth, replication, and preparing for that division. So it's G1S and G2. If I were to draw the cell cycle, which I'm gonna, so I would put G1, S, G2, and M. I'm gonna draw a circle around it, separate it into 4 parts. Now, I'm going to draw an arrow from G1 to G2 And I'm going to label that as
7.
interphase. All of those are a part of interphase. Be sure you put this drawing in. That is important. You often have to interpret the cell cycle with drawings on state and local exams. So the only thing that's not in interphase is what?
Mitosis.
Yeah, mitosis is the only thing not included in interphase.
What's the point of the other phases then?
Really, the point is to get ready for mitosis.
Like 3 other phases just ready for mitosis?
Yep. So G1, we're resting after mitosis. S, we're copying the DNA in the nucleus, so we have 2 sets to get ready for mitosis. G2, other things are happening within the cell to prepare for mitosis, and then M is mitosis, which we'll go over in detail in just a second.
Okay.
All right, are we good? Everybody ready to move on? All right. So here's that same drawing. This one's a bit more detailed. You don't have to draw it right now. Just want to give you kind of an idea of what to expect. New heading, new ink, left-hand column. We're going to G1. I want you to note that the interphase is happening during G1. So G1, interphase happening. Here it grows. It makes proteins and organelles. Centromere and centrosomes are made. I know these vocab words are not things you're accustomed to right now. We're just setting the foundation. We're going to dig into what these words mean here shortly. Not today, but soon. So today, just get it down. Later on, it will make a bit more sense. And then you're also going to note if this checkpoint is missed, what do we get?
Cancer.
Yes. Remember another word that the state may use to refer to cancer. They may just say tumor. You can kind of use those words interchangeably, okay? So G1, interphase is happening. They're growing, making proteins and organelles. The centromere and centrosomes are made. If we miss this, we can get cancer or a tumor. Not a ton of writing here, so I'm going to give you just a little bit longer.
What if you miss mitosis?
Everything stops. Like if cells can't be copied, so cells have a limited lifespan.
Can I go to the bathroom?
Is it an emergency or can we wait? Because missing notes is tough.
Actually.
Okay.
Don't we get like student notes in that thing over there?
No, but you still have to take them.
Oh, that's good. I mean, yes, we do.
All right. New heading, new ink, left-hand column. This is S. Again, interphase is happening. This is where DNA replication happens. It is copied from one strand to form 2 identical strands. It unzips up the hydrogen bonds. There's actually an enzyme that does that, and it is called helicase. It unzips using helicase up the hydrogen bonds. Helicase is an enzyme, which is why we started this week with our enzyme foldable, so you can understand how those things work. Again, multiple combinations of the 4 nucleotide bases code the instructions in our DNA. All organisms use the same genetic code, so the genetic code is a universal code. Genetic code is a universal code.
So what happens whenever your body stops making cells?
Eventually, every cell in your body would die, and there'd be no new ones. You'd die.
You just drop.
Well, it wouldn't be fast. It would be slow and painful.
Does that happen?
No. Couple more seconds on this, and then we're going to go into adding a drawing into our notes because I really want you to understand what DNA replication looks like. DNA replication happens in S phase, copied from one strand of DNA to form two. It unzips with helicase. Now, what I'm about to give you is a simplified example. There are more steps in it. We're not going to dig into the specifics right now. I just want to give you a brief overview so that later you're prepared for specifics. Yes, ma'am?
Yes, ma'am.
All right. Okay, so you're still in your notes. Here's what I want you to draw. You're going to draw first. So that's the sides of DNA. Those are my phosphate bonds. I'm going to put my rungs down the middle. These are my bases joined by hydrogen. This is a DNA double helix. Draw this in your notes. This double helix can be ripped up the center by an enzyme called helicase. That helicase takes those hydrogen bonds up the middle, and it's just like unzipping a zipper. It's going to pull that DNA apart into two sides. So it's like two halves of the ladder, and the ladder's been cut down the middle. So we're going to go from here to here. Now I have two halves of DNA. Does everybody see that? Each one of these halves is going to become one part of the next step. So that's what I had, right? Let's say on this side, let's say it's A, T, C, G. And this side would be T, A, G, C. All right? Now, a whole bunch of other enzymes come in to do a job, okay? We're not going to go into those details right now. But what they do is they come in and they're going to build another section to match both of these. So it's going to, see how I change my ink? That's to show you that the other thing is new. So if you want to change your ink here, that may help you remember as well. So it's going to come in and it's going to throw here, I'm going to use my black again. It's going to put a T, it's going to put a G, and a C. And then here it'll put a T, an A, a T, a C, and a G. So these two are now both new strands. Do you see how half of each new strand is old? It's from the original DNA. And half of each new strand is new. It was just built, wasn't it? My Lord. So get this down. Miss Stoley. Yes, ma'am. Guys, I gotta jump off the screen. I need my attendance.
Yeah, do your attendance.
Are we missing anyone?
Yeah, Alex.
she's not here. Rihanna?
She's right there.
Okay. I understand you may not have finished that drawing perfectly. It's okay. You get the gist. All I want you to know from that, this is kind of the next step, this would be step 3, is that they then recoil. And I now have 2 strands of DNA, right? One side is from that old strand and one side is new and it was just built.
Wait, so does that happen every time?
It happens every time a cell goes through its cell cycle.
So it's basically like
It's cloning itself.
but it's not.
All right, so we just finished talking about S phase. I do want you to list the enzymes here. Like I said, we're not going to go into great detail about the process yet. We will later, but I want you to get them down. Helicase unzips. Polymerase builds. Primase initiates. That's a fancy way to say starts. And ligase glues. Polymerase only works 5' to 3'. And the leading strand replicates continuously. So that first strand, the black one I drew, would be our 5' to 3', and it would copy it continuously, okay? We'll clarify 5' to 3' and 3' to 5' at a later date when we are looking at a model. For now, just get it down in your notes so you have it to reference later. Jot it down, doesn't have to be perfect. A bit. We struggle in 3rd to finish because the announcements take up so much of our time.
It's gonna go into next Monday.
If we can get through, I'm hoping to finish it today. But we're gonna have to push hard. So I don't want to do another day of notes. All right. So we are still in S phase. I want you to know that you have the leading strand and the lagging strand. The lagging strand replicates in smaller pieces. These are called 3' to 5' lags, and they are chunks on the lagging strand. We call these Okazaki fragments. Again, none of this makes any sense to you right now. That's okay. Get it down. I'm going to clarify this for you later, and I promise it will make sense then.
Is it named after a sign?
It's the guy who discovered it, yeah. Okazaki fragments. This is when 3' goes to 5' during replication in S phase. Leading strand and lagging strand. Lagging replicates in smaller pieces. It's your 3' to 5' strand. It's replicated in chunks. We call those fragments Okazaki fragments. All right, I'm about to move on. No? Do I need to wait? For those of you who are done, I can just go ahead and put here what you need to know. Any mistakes during S phase can equal what we call a mutation. You know where to put it? In the crate, probably. Or you want to put it in your stuff?
Probably not.
Yeah, put it with my stuff. Any mistake during S phase can equal what we call a mutation. We'll delve more into the types of mutations in a later unit. All right. We got to get through mitosis today. We're going to skip that. I'll tell you that we call this semi-conservative. You don't have to write it down. It just means we have one old and one new, just like I showed you in our example.
Isn't that like, you said different things come in and make a difference during, wouldn't that be like?
It's a new one.
Hereditary?
So you're thinking meiosis, which we have not talked about yet. There's a similar process that makes the DNA that gets passed to children. This is not it.
Okay.
All right. So I just want you to see this image. This is an image representing what DNA replication looks like during the cell cycle. You don't have to draw it. In your notes, new heading left column, I want you to put G2 interphase happening. This is rapid cell growth. Protein synthesis, prepare for mitosis. If checkpoint is missed, we get cancer. Rapid cell growth, protein synthesis, prepare for mitosis. If checkpoint is missed, we get cancer. We are close to done, guys. Rapid cell growth, protein synthesis, prepare for mitosis. If missed, we can get cancer. Give me a fist to 5 on this, non-verbally. Show me with your hand. How much longer do you need? I see a 1. I see 3 ones, so we'll hang out just a second. All right, here we go. New heading, new ink, left-hand column. Here's the big mamma jamma. Mitosis, nuclear division. This is my M phase. This is after interphase. We are dividing the nucleus. There are 4 phases. It is prophase, metaphase, anaphase, and telophase. prophase, metaphase, anaphase, and telophase. I like to use the acronym PMAT. PMAT. I remember that M is middle and A is away. PMAT. P is in the middle, A is away. We're going to break each of these steps down here in a second into what is happening at each.
You don't need to move your either to right.
So what I'd like to do is when I say PMAT, I would like for you as a class to say prophase, metaphase, anaphase, telophase. Can we handle that?
No.
Yes.
No.
Yes.
No.
Yes. So I'm going to say PMAT. What are you going to say? Okay, we're going to do it again. I say PMAT, you say prophase, metaphase, anaphase, and telophase. Let's do it. PMAT, prophase, metaphase, anaphase, and telophase. PMAT, prophase, metaphase, anaphase, and telophase. PMAT, prophase, metaphase, anaphase, and telophase. All right. We're still talking about mitosis. So there are a few things that happen in mitosis. We coil the DNA into chromosomes. This is what happens when we're ready to do mitosis. We line those chromosomes up, we separate them, and we have 2 new nuclei. Coil up the DNA until those chromosomes are visible. Like yesterday, we talked about twisting hair. If you've ever had experience with twisting hair, if I twist it enough, it actually tightens and pulls in on itself. That's exactly what's happening to a strand of DNA when it is forming a chromosome. Those chromosomes then line up in the middle of the nucleus. After they're lined up in the middle, they get separated and pulled away from each other, and then they're at opposite ends. So prophase is right about when everything is about to begin. So when I say prophase, we're going to make a hand signal like this. The nucleus is just kind of there ready. Okay? In metaphase, we line up in the middle. So when I say metaphase, we're going to make a hand signal like this to show they're in the middle. Are you looking?
Yes.
Anaphase, they start to move apart and they kind of look like A's moving apart. So we're going to do this. And then telophase, we've got almost 2 completely separate nucleuses. So it goes prophase, metaphase, anaphase, telophase. You got it? So let's try it together. We're going to do prophase, metaphase, anaphase, telophase. Again, prophase, metaphase, anaphase, telophase. Okay? So we're going to look at that in picture form now. Give me a fist of 5 if you're still writing here. 1 more minute. That's fine. I'm not going to make you draw the steps in your Cornell notes today because we are actually going to do some guided notes together next week where we get to draw them. This is what it looks like. You don't have to draw them, but remember it's prophase, right? Do it with me. Prophase, metaphase, anaphase, telophase. Do y'all see how that top picture looks like that?
Yes.
All right. Last step, left-hand column, cytokinesis. The cytoplasm divides, it cuts in 2. The cytoplasm divides, it cuts in 2. So, here's what I want you to put. We end up with 2 identical clones with same genes. When you are done writing that, you can fold your Cornell notes in half and put them back in the front pocket of your journal. We have 3 minutes until the bell. You are welcome to go ahead and clean up your journal boxes, your pens, and get ready to go. Before we exit, once everyone is clean, I am going to ask you about our exit ticket once again.